Hello students, welcome to online classes. In the last class, we discussed about the drugs and the classification of drugs. In today's class, we shall discuss about the drug-target interaction. Before starting this topic, shall recap the meaning of drug. What is drug? Drug is a chemical. When we intake, it interact with the biomolecules in our body and produce a biological response. So, in order to function biochemical reactions in our body, biomolecules plays a prominent role in our body. Here, we find so many biomolecules in living systems. Do you think all the biomolecules perform same functions? No, not at all. Each biomolecule have its own specific function in our body. Let us know about the function of biomolecules one by one. I am going to take the first one, proteins. This proteins is also called as enzymes. What is the function of this enzymes in our body? It acts as a biocatalyst. What is this catalyst? Catalyst is a chemical substance which helps in changing the speed of a reaction. Either it increases the rate of a reaction or decreases the rate of a reaction. In laboratories, you add some catalyst in order to change the rate of a reaction, isn't it? In the same way, even in our body, these biomolecules require some catalyst in order to increase or decrease its biochemical reactions. And the catalyst used for biochemical reaction is known as biocatalyst. What is the function of enzyme in our body? It acts as a biocatalyst. What can biocatalyst do in our body? It increases or decreases the rate of a reaction. Okay. Second one, carbohydrates and lipids. Where do we find these carbohydrates and lipids? These are generally found in foodstuffs. What is the function of carbohydrates and lipids? These involved in the structural process of a cell. Structural process. Each cell requires a carbohydrate and lipids to get a particular structure of a cell. What is the function of carbohydrates and lipids? It helps in a structural process of a cell. The next one is nucleic acids. What is the function of nucleic acids? The function of nucleic acid is it transfers a coded genetic information to a particular cell. We find billions of cells in our body. So, do you think all the cells carry a function in our body? No. This nucleic acid will transform information to a particular cell. This is the function of nucleic acid. The next one is receptor. These receptors are also a kind of protein. These receptors are also a kind of protein. What is the function of receptor? These receptors will involve in communication system throughout our body. How it perform a communication system in our body? It transform message from one nerve cell to the other nerve cell throughout our nervous system. What it do? Involves in communication system. Involve in communication system. So what receptors will do? It involves in communication system. How? It transfer message from one nerve cell to the other nerve cell throughout our nervous system. So where does this receptors found in our body? These receptors are embedded in cell. Consider this is cell, and on this cell membrane receptors are present. Receptors. What does these receptors will do? These receptors will transform a message into a cell. Suppose a chemical message is coming near the cell. Receptors are present on the cell membrane, and these receptors will leave the chemical outside only and transfer the message inside the cell. This is how receptors act as a communication system in our body from one nerve to other nerve throughout our nerve. Discuss about the drug target interaction. Under drug target interaction, we find two categories. The first one is drug enzyme interaction and the second one is drug receptor interaction. First one, drug enzyme interaction. The second one is drug target interaction. Under drug target interaction, we find two categories. First one, drug enzyme interaction. That is the interaction between the drug and the enzyme in our body. And the second one is drug receptor interaction. That is the interaction between the drug and the receptor in our body. First, we shall discuss about the drug enzyme interaction. Before knowing about drug enzyme interaction, let us know about the enzyme, that is which act as a biocatalyst, its activity in our body, that is catalytic activity of an enzyme. How how the enzyme catalyze the biomolecule in our body. Let me know about it in detail. This catalyst performs two major functions in our body. The first one is this catalyst will bind the substrate to the active site of an enzyme. What this catalyst will do? This catalyst will hold the substrate to the active site of the enzyme. Then what it will do? This catalyst acts as a This catalyst will hold the substrate on a enzyme. And the next is this catalyst provide a functional group to the substrate in order to carry out the chemical reaction. Let us discuss it through a diagram. Suppose this is an enzyme present in your body. This is the enzyme present in your body. Then a substrate. What is meant by substrate? Substrate is a chemical compound which undergoes in a biochemical reaction. Biochemical reaction ನಲ್ಲಿ ಸಬ್ಸ್ಟ್ರೇಟ್ ಅನ್ನೋದು ಒಂದು ಕೆಮಿಕಲ್ ಕಾಂಪೌಂಡು, ಅದು ಬಯೋಕೆಮಿಕಲ್ ರಿಯಾಕ್ಷನ್ಸ್ ಅಲ್ಲಿ ಇನ್ವಾಲ್ವ್ ಆಗುತ್ತೆ. ಹೇಗೆ ಇನ್ವಾಲ್ವ್ ಆಗುತ್ತೆ? ದಿಸ್ ಈಸ್ ಎ ಸಬ್ಸ್ಟ್ರೇಟ್. This is enzyme and this is substrate. This substrate will be in search of active site of the enzyme. This is the active site. When it gets the enzyme having a similar shape, then it will bind to the active site of the enzyme. This is having the similar shape, isn't it? When it will bind to the enzyme, the orientation fits in a proper way. That is like this. This is an enzyme and this substrate will come and bind to the active site of the enzyme. Binded on the active site of the enzyme. How it binds on the active site of the enzyme? Through dipole-dipole movement, ionic bond, hydrogen bond, or van der Waals force of attraction. Among these, with some bonds, it binds to the active site of the enzyme. Then what it will do? After binding to the active site of the enzyme, the next function performs. What is the next function? This is the first one. Second one. Substrate binded to the active site of the enzyme now, and if this enzyme consisting of a functional group, there is some functional group here, and this functional group attached to the substrate. Functional group major functions of a catalyst in our body. What it will do first? A substrate present in our body, that is a natural substrate present in our body, this will come in search of active site of the enzyme. Active site is nothing but it will come in search of similar shaped enzyme in our body. We find many enzymes in our body having different shapes. So among those different shapes, it will be in search of the similar shape of the enzyme. When it finds a similar shape of the enzyme, then it will bind to the active site of the enzyme. It will bind to the active site of the enzyme through ionic bond, dipole-dipole interaction, hydrogen bond, or van der Waals force of attraction. Van der Waals force of attraction. Among these, with any one property, it will bind to the active site of the enzyme. Then, if the enzyme consisting of functional group, then that functional group will attack to the substrate and the reaction is carried out. Chemical reaction carried out. This is the two major functions of a catalyst in our body. And note the point, we have different shaped enzymes in our body and hence the substrate will be in search of similar shape. If the enzyme is having a shape like this, and the substrate will come in the search of the enzyme, and the substrate having a shape like this. Is this possible to bind here? No, because this cannot properly orient on this enzyme because the substrate is having different shape and the enzyme is having different shape. But here the substrate and the enzyme both are having similar shapes so that it can properly orient on the enzyme. Has these are weak bond so that some pressure applies on the enzyme inside our body. From all the sides pressure applies, and for example, if a glass is broken and you will stick it by using some gum and keep aside, but again it fell down and break. What it happens? The place where it has broken before, in the same place it breaks first. In the same way, here there was a gap at first at the side and the substrate binded to the enzyme. So these are the pieces added. So when the pressure is applied from all the sides, then this portion will get high pressure and the bond between them, that is dipole-dipole moment or hydrogen bond or van der Waals force of attraction, this will break and forms an intermediate and form an intermediate. These intermediates do not have any participation in the reaction, but it will give a pathway to the further chemical reactions. And this intermediate ಈ ರಿಯಾಕ್ಷನ್ಸ್ ಯಾವುದೇ ಒಂದು ಪಾತ್ರನ ತಗೊಳ್ಳೋದಿಲ್ಲ. ಆದರೆ ಈ ಇಂಟರ್ಮೀಡಿಯೇಟ್ ಏನು ಮಾಡುತ್ತೆ, ಫಾರ್ಮ್ ಆಗಿರುವಂಥ ಇಂಟರ್ಮೀಡಿಯೇಟ್ಸು ಆ ಇಂಟರ್ಮೀಡಿಯೇಟು ನೆಕ್ಸ್ಟ್ ಕೆಮಿಕಲ್ ರಿಯಾಕ್ಷನ್ಸ್ ಆಗೋದಕ್ಕೆ ಒಂದು ರೂಟ್ ತೋರಿಸುತ್ತೆ. ಸೊ ಇಂಟರ್ಮೀಡಿಯೇಟ್ ಒಂದು ಪ್ರಾಡಕ್ಟ್ ಆ್ಯಂಡ್ intermediate will form. If these is good, then the chemical reactions. Under drug target interaction, we shall discuss drug enzyme interaction. What happens in catalytic activity of an enzyme? An enzyme is there and the substrate will come in search of an enzyme having the similar shape of the substrate. This is the substrate. It will come in search of an enzyme having the similar shape. Then, after finding the similar shape of the enzyme, it binds to the enzyme by hydrogen bond or ionic bond or van der Waals force of attraction or dipole-dipole interaction. After binding, a pressure is applied on the enzyme and the substrate in our body. Then what happens? This enzyme is broken into an intermediate the substrate is broken into an intermediate and product. Now it's important for the next concept. If the product form is favorable to our body, then it is fine. The enzyme will be attached to the other substrate in the coming reactions. If the products are favorable to our body, then these products will be in search of other enzymes. But if these products are not favorable to our body, then we feel sick. So that is why we feel ill sometimes. Why we feel ill? Because these products will not be favorable to our body. By that time, what happens? We consume drug. This is what now drug enzyme interaction. After consuming drug, how this drug interact with the enzyme? The drug interact with the enzyme in two ways. The first one is this is an enzyme and this is the natural substrate present in our body and this is our drug. The substrate which is supposed to attach to the enzyme, but this drug will compete with this substrate and attach to the enzyme. This substrate wanted to come and attach to the enzyme, but what this drug will do? This drug will move fastly and attach to the enzyme. Where it will attach? This is the active site, right? It attached to the active site of the enzyme. This is the drug. This is the substrate. So what happened now? The drug compete with the substrate and attached to the active site of the enzyme so that it blocked the active site. There is no space for the substrate to attach to the enzyme now because this drug blocked the enzyme, and the drug which compete with the substrate and attached to the enzyme is known as competitive inhibitors. Competitive inhibitor, and after competing and attaching to the enzyme, it will inhibit the attachment of a substrate to the enzyme. So, it is also known as enzyme inhibitors. Enzyme inhibitors. Why it is the drug is called as competitive inhibitors? Because it compete with the substrate and attach to the enzyme, and hence it is called as competitive inhibitors. By attaching to the enzyme, it will inhibit the attachment of a substrate to the enzyme, so it is called as enzyme inhibitors. This is the first case. And the second type is, there is an enzyme and it is our natural substrate enzyme. This is the active site of the enzyme. But when the drug comes inside our body, that drug, instead of attaching to the active site, it will attach to some other site of the enzyme. Attached to the some other site, and that different site is known as allosteric site. Allosteric site. Allosteric site. After attachment of a drug to the allosteric site, this will block the enzyme and change the shape. Or oriented like something. This is the shape of the enzyme after the drug attached to the allosteric site of the enzyme. And then this substrate can't able to attach to this shape because substrate is having this shape but the enzyme is having this shape. It can't orient properly here. Then this substrate will keep in search of other enzyme. But here what the drug will do, if the attachment is through a covalent bond, then the bond will be strong and it will not break. Here it broke. Here it broke and formed an intermediate product. But here, because of strong bond, that is covalent bond, the bond is not broken. Then this enzyme is degraded from the body, that is ejected out from our body. This is how the drug interact with the enzyme in our body, that is drug enzyme interaction under the main heading drug target interaction. Let us recall it now. What happens first? The drug target interaction. In the drug target interaction, the first one is catalytic activity of the enzyme. What is the activity of the catalyst in our body? There is an enzyme in our body, and to this a natural substrate comes and bind to the enzyme. If the enzyme and catalyst, if sorry, if the enzyme and the substrate having a similar shape, then it will hold on the active site of the enzyme through ionic bond, hydrogen bond, or dipole-dipole interaction, or van der Waals force of attraction. After binding, the force applied on the enzyme, then that substrate is broken into intermediate and the product. If those products are favorable to our body, then those substrate will be in keep on search of other enzyme which is suitable for its shape. But in case if those products are not suitable for our body, then we feel sick. And when we feel sick, we take a drug, and this drug will compete with the substrate and attach to the enzyme and block the enzyme, so that this substrate could not able to attach to the enzyme. The competition between the drug and the substrate, and thus it is known as competitive inhibitor. Why it is called as inhibitor? Because it blocks the enzyme that prevent the substrate to attach here, and so that it is called as enzyme inhibitors. This is in first case. And if the drug attached to the enzyme in different site instead of active site, then that site is known as allosteric site. After attaching to the allosteric site, the shape of the enzyme changes so that the substrate could not able to attach, and it will be in search of other enzymes. If the attachment of a drug and the enzyme having a strong covalent bond, then this bond could not be broken. Those enzymes are rejected out from the body. This is about drug enzyme